Up our next speaker, Dr. Evan Snyder, MD, PhD. He's the professor and founding director, Center for Stem Cells and Regenerative Medicine, founding director, Stem Cell Center and Core Facility, founder and director, Stem Cell Training Program at the Sanford Burnham Prebys Medical Discovery Institute. Now, I've known Evan for years. He's a pediatrician by training. He's taken care of many little children, and his heart is pure, his aim is true, and I've run out of adjectives because it's late in the afternoon. Evan Snyder.
You're right, I'm a pediatrician, so I'm going to be talking about the complete opposite end of what Mahendra's talking about. In fact, I'm going to be talking about whether or not we should be treating diseases of adulthood, maybe even diseases of aging, but in infancy, in the prenatal period. Now, there's no question that if you start doing that, okay, hopefully this will start working, that there's going to be ethical challenges, and I think we'll get to that, I think, during the Q&A. Now, the one thing I'm going to warn you is that Bernie said this is public day, which meant that the expectation was that people would be coming off the street who may not know anything at all about stem cells. So I apologize to those that are really au fait with stem cell biology, real mavens, if I start a little bit simplistic and then I don't mean to insult your intelligence or your knowledge, I just want everybody to kind of be on the same page. And of course, Einstein did say that if you really understand what you're talking about, you should be able to explain it simply. So for those not familiar with stem cells in the audience, this is a pediatrician's view of stem cells. Now, we can superimpose it on what we know. And Bernie also did mention I'm also a neuroscientist, so a lot of what I'm going to be talking about, except for one aspect, is going to be in the nervous system. Now, the most important thing, I think, with regard to this topic is to recognize what stem cells are. Stem cells are not there to be entrepreneurial. Nature put stem cells there to perform two roles. One, put the system together and keep it going, organogenesis and then homeostasis. And our job, if we want to engage in regenerative medicine, is to learn how to first learn what those programs are and then learn an intelligent way to reinvoke them or exploit them. Now, as we all know, there's many ways to get stem cells. You certainly can get it from the blastocysts. We've been doing that in humans for over 20 years now. And of course, one takes the inner cell mass, cultures that out, and you can get the vast array of cells of the body. Sometimes we talk about implanting them. And then, of course, again, about 15 years ago, we recognized that this direction was not unidirectional, that we could actually go back in developmental time, which I thought was mind-blowing, but has actually become probably one of the most important concepts in developmental biology. Either way, you can get all the various cell types that characterize the body, including the nervous system. The reason I'm bringing this up is to make the point that no matter how you choose to get a neural stem cell, whether you take it from a fetus, you take it from an adult, you take it from the inner cell mass, you make IPS lines, the biology ultimately is the same. We still have the same biologic guardrails, the same biologic imperatives that we need to adhere to, which means the stem cell is there to put the system together. In the nervous system, it's not just neurons, it's astrocytes and oligodendrocytes that work together. And there's a dynamic. If you're going to do a transplantation with a stem cell into an abnormal host, there's a dialogue, and our job is to understand all of these various arrows. And the arrows are multidirectional, including stem cells talking to each other. When they talk to each other, there's a division of labor. You do this, I will do that. One of the roles is actually the non-neuronal cell that engages in something that we called many years ago the chaperone effect. It's the stem cell derivative talking back to the host. It does it by, it gives it neuroprotective signals. There's metabolic and housekeeping factors that are taken up by the receiving cell. There are bits that fulfill an anti-inflammatory function, an anti-scarring function, that mobilize endogenous neural stem cells that make those send out neurites that cause pro-angiogenesis in the host. And they do it by diffusible factors, and some of those are our well-known neurotrophic factors. They do it through cell-cell contact, and I'll talk about that a lot. That can be through gap junctions, tunneling nanotubes, fusion. Lately, we've learned that they do it by budding off microvesicles and exosomes. At the end of the day, even if you don't care about the developmental biology, even if you simply care about making things better, I'll argue you're still a translational developmental biologist. And you have to recognize that diseases, injuries, are not isolated. When something happens to the brain, it's just not nerve cells. It's vasculature and extracellular matrix. And sometimes these depend on each other, both for the function, their good function, and also for the disease. And most importantly, no matter how we try to delude ourselves in thinking that we know how the brain or the system is put together, we don't. And the complexity of the nervous system is, I would say, a century away from our completely understanding how that is done. Therefore, it probably makes more sense to preserve what you already have and try to do that the best you can. Now, that means you have to be able to predict disease and disability. What that means is disease modeling. And disease modeling gets us into personalized or precision medicine. Now, one way, and I think it's been advanced a great deal by the recognition we can make IPS lines, are diseases in a dish. The way we used to start out, one of the ways we started out was take normal cells and assault them in ways that we thought a disease took place. With IPS lines, of course, we could start having disease models. And if you have a disease model, in other words, disease in a dish, a cell, you can understand or at least try to study the underlying mechanisms, look for fingerprints, diagnostics, prognostics, biomarkers, drug targets, then identify the drugs against those targets, and try to look for agents that can cause compensation. Now, as I say, we began doing this before iPS cells came along, but it was tough. It was a slog. We would take neural stem cells, let's say, out from diseased fetuses or miscarriages. We would pull them out from autopsy specimens of patients that had died in adulthood. Or we would make ES cells from discarded blastocysts that had been diagnosed as having a disease but would never be implanted, and make ES cells from that. But the advent, of course, of IPS lines meant that we could start taking inaccessible cells from living patients and all of their relatives. And very few of my relatives I know would donate their brain while they're alive. But this opened up all kinds of opportunities for doing even better disease modeling. You could have populations that could be studied. And the conceit back then, and this seems to have been borne out, is that when you make a neuron, let's say, from a patient with a particular disease, these neurons, strange as it may seem, in most cases have that disease, or at least seem to have a phenotype that allows us to better understand the disease. Well, if we want to start doing this on a population-wide basis, we want to start knowing who to fix and when, we actually need a library of molecular and functional data on every person, ideally on the first day of life, maybe even prenatally. Well, how are you going to do that? Well, I thought about it a lot, and I'm going to propose one example. And again, as Bernie mentioned, I'm a neonatologist. And one way to do that is to access something that every single baby has when they're born, straight out onto the warming table. They all have an umbilical cord. And I'm not talking about the blood. This little piece over here, anybody who's been in the delivery room knows that little chunk of cord is cut and gets thrown away. It's obtained non-invasively. In fact, it's biological waste. Well, we figured out that you could take that piece of cord, dissociate it, and turn it into IPS lines. Once you have them into IPS lines, the key is you can not only get a molecular fingerprint on those cells, but now you get them into cell types that have different functions. So now you have a phenome, a phenotypic marker, not just a molecular marker. And the key, anybody who's a clinician will know this, the key is not having a marker. The key is correlating that ongoing throughout life with clinical correlation, because there's a huge, vast array of normal. Finding data is not the hard part. Finding meaningful, clinically relevant biologic data, that is the hard part. And what we can start doing is having this library and start correlating it as we move forward with how does a baby do. When we have this bank, and my argument is this should be what we obtain the way we get footprints on newborns. It should be part of routine obstetrical care. First, you can start identifying the babies that are at risk, knowing what's going to go on, figuring out, if you do find a problem, what drug will this particular baby be most responsive to, and what will the prognosis be. But the other conceit, and I admit it's a conceit, is that maybe we can have a crystal ball. Not immediately, but as we continue going prospectively in a non-cherry-pick kind of fashion, clinical correlation. So which baby is going to get autism at 2, or diabetes at 5, or a brain tumor at 10, or MS at 24, pancreatic cancer at 40, Alzheimer's at 65? Then the next question would be, once you have this, what's most predictive? Is it the transcriptome, the epigenome, the proteome, the metabolome, the connectome? And lately we've been focusing on organoids. These are 3D representations of the derivatives of IPS lines. Now I'm going to shift actually away from the brain into the lung, because this is actually kind of cool. I have a neonatologist in my group, Sandra Leibel, who's made mini lungs. And I think they actually look like pretty good mini lungs. And what you do is you take skin from a patient or blood, and you go through the normal reprogramming that you're all familiar with. Now the good thing is, and this is the reason I'm bringing this up for the lung, is that you can take them from patients of different sexes, different genders, different racial backgrounds, which becomes important because now you also have patient-specific mini lungs that can represent a patient who grows up in a deprived environment, that's exposed to toxins, an African American who we know perhaps gets sicker when exposed to COVID. So this was kind of cool. Let me just show you how these mini lungs were pretty amazing. They respond to dexamethasone, they have little respiratory movements, they have cilia, and the cilia will move mucin around. And of course, we did this during the pandemic and infected the mini lungs with SARS-CoV-2. I won't go into the details of that except to say that we did it to find drug targets. We identified drug targets, we identified drugs, and unfortunately we actually found that there was some disparity in how different racial and ethnic groups responded to A being infected by SARS-CoV-2, and then responded to the drugs. I won't go into that except to say that talk about predicting, once we have this lung, can we start actually figuring out, as a little crystal ball is, what should we look for patients that might start getting lung cancer? Remember, we've emulated lung development. We've started at the epiblast, and now we've moved all along into at least a baby lung. So we started interrogating is that me popping? I guess, sorry. Interrogating the lung to see, could we induce cancer and then see the cell that would start giving rise to cancer starting from normal? One way to do that, of course, is to put in known or candidate mutations. Another is to actually take cancer, push it back into a normal IPS line, precancerous state, like it's in the epiblast, take your foot off the brake, and let cancer reemerge. So that's what we're doing to crystal ball what goes on in the lung. I'm a neuroscientist, so I have to go back to my sweet spot, which is neurologic disease. You can do the same thing with neurologic disease. And of course, organoids there are these 3D accumulations of multiple cell types that have and develop the normal cortical layers. And if we took IPS lines, let's say from a baby with lissencephaly, small brain, Miller-Dieker syndrome, I think you can see why it's called small brain over here. There's a Miller-Dieker kid, and here's a normal kid. Well, here's what the organoids look like. And it's kind of astounding that even the organoids seem to be smooth in comparison to the normal cerebroid. Now, that doesn't tell you what to do, except when we start, hopefully this is playing, when we start looking as to why that developed, it looks as if simply the cells do not migrate in the inside-out way that they normally should. As you can see on the left, they just all cluster right around here. Well, that can become a drug target. And if we can diagnose it early, we can intervene early. Apropos to what Mahendra was talking about, can we do the same thing with aging? And what's kind of interesting, and Mahendra talked about both supercentenarians, these are the elderly over 110 years of age, and he talked about progeria patients. This is slow aging in the supercentenarians, very rapid aging in the progeria patients. Well, making an IPS line starts everybody up at the starting gate. It erases most epigenetic marks of aging, but there's something intrinsic about the speed of aging. I don't have the answer, so don't wait with bated breath for an answer. But if we let these start moving forward and differentiate them and see what the key differences are, maybe we will come up with some key markers of what mediates slow or rapid aging and wellness into old age. So now that you know, and I put "know" in quotation marks, what are you going to do about it? And the key is treat as early as we possibly can. Now, what does that mean? Well, from a stem cell biologist, it means trying to get fetal neural stem cells. And I'm going to show some of the data. Some of it will be mouse, and those will be blue. Some of them will be human. And quite a while ago now, we isolated some human neural stem cells that we decided we would use not only to study development, but to see whether we could treat. So the first part of treating early is treat pre-symptomatically. Now, the key to that might be in a disease like ALS, where it hits somebody young, and in Lou Gehrig's case, of course, he didn't know he was going to get it. But there are enough familial cases of ALS where you could intervene early. There's some trials out there now. This was a stem cell trial that we did with the SOD1 mutant mouse. This was an animal that was treated pre-symptomatically early. He's walking a rotor-rod, which is pretty remarkable because at this point in time, this animal should be dead, not trying to walk a rotor-rod. We didn't replace motor neurons, but what we did do was protect the motor neurons. We changed the toxic astrocytes of the host so that they were now kinder, gentler astrocytes by changing the neural stem cell repertoire of the host and then supplying our own trophic and supportive gray matter oligodendrocytes and astrocytes. Now, we treated pre-symptomatically, but the mice ultimately did die much, much later, maybe more than a year later, but they ultimately died. So we treated in adulthood. Maybe that was too late. So maybe we should move earlier. How about day of birth? And we can do this. We can do this in mice. We can actually do this in babies. Here we're injecting neural stem cells into the ventricles. It's a really simple bench procedure. In babies, it's just as easy. You stick them into the ventricles. As most of you know, they integrate in the subventricular zone and then migrate out to the developing brain. So wherever you see blue cells, those are donor wild type cells. And you can see them because they're expressing a gene, in this case, LAC-Z. So obviously, they're integrating gene-expressing cells. Here it's LAC-Z, just a reporter gene, but imagine that it was something therapeutic. Now, what's cool is you don't have to engineer these cells. You can if you want, but because these are normal cells, they produce normal lysosomal enzymes, normal housekeeping genes, so that you can bring that in early on in a Trojan horse fashion, which is something that we demonstrated many years ago for mucopolysaccharidosis, where it was kind of, think of it as a bone marrow transplant to the brain, where we could now distribute, and you only need a little bit of enzyme, distribute it throughout the brain, and it eliminates lysosomal storage. So that's expression, global expression of a gene product. What about global expression of a new cell or a replacement cell? You can, as long as you stay consistent with the biology and with development. So maybe not a cell that's born prenatally because we're going day of birth, but oligodendrocytes are born postnatally. So can we replace myelin? That's what that's what oligodendrocytes do. And for those not familiar with the neuroscience, myelin is an insulation. Those who are really familiar will know that this schematic is completely screwed up because that's not the way the impulses go. They actually go from the cell body out. But it was such a great video, I couldn't avoid not using it. So what we did do is go into an animal called the shiver mouse. This is a dysmyelination, not a demyelination. Oligodendrocytes do not form well. So you have a shivering mouse, as you can see. You go in day of birth. Even though this is not a disease that doesn't manifest itself until adulthood, now you can see an animal. Even from this view, you can see the shiver is gone. It's quantified, but I won't have time to show you. We can do it in other white matter diseases like Krabbe's disease, which is a Gaucher deficiency. Here's the Twitcher mouse model of Krabbe's disease without any oligodendrocytes, transplanted day of birth. You can see oligodendrocytes that now create myelin. Now, let's move to neurons, because that is a little bit interesting. So this is a mutant called the nervous mutant. Purkinje cells degenerate. Purkinje cells are born in mid-gestation, not day of birth. Other things are born in the cerebellum, but not Purkinje cells. And what happens in this mutant, so Purkinje cells look like a pearl necklace right here in a single monolayer, and they're just these beautiful multi-branch cells. And you can see at adulthood, here's a big full-size wild-type cerebellum. Here's a nervous cerebellum. You can see it's much smaller, but more importantly than being smaller, as opposed to this necklace of good Purkinje cells, most of these at adulthood are completely gone. However, and this doesn't happen until adulthood, however, on day of birth, before there's any evidence that there's something going on, you transplant the neural stem cells into the cerebellum of the newborn nervous mouse, and now at adulthood, the necklace is there. We've not replaced the necklace, the Purkinje cells. What you can see over here is that the green donor cells have arrayed themselves, and they're sending out these processes to the host Purkinje cell. And you can almost see these are little gap junctions that they're forming. It's almost embracing and hugging the Purkinje cell. And through gap junctions, it's restoring metabolic function. I won't go into the details of the molecular system, but this happens in many Purkinje cell degenerations. Here's an SCA1 mouse, same story, decorated by gap junctions from donor wild-type cells. So how about if we start going extremely early, even earlier? Now, can we go into cancer before it's cancer? This is a mouse model called the GRAS model, and it inevitably at adulthood develops a brain tumor. And this is what a mouse looks like. Around here, around the ventricles, is the home of the neural stem cells, the adult neural stem cells, and what they should do is migrate out to the olfactory bulb. In this animal, what's kind of interesting is it starts developing a brain tumor, and the cells that should normally go out to the olfactory bulb are instead pushed and contribute to the tumor over here. However, if we know that this is happening, if you start a drug that changes this signal transduction, inhibits this pathway, now not only do the cells start moving back out to the olfactory bulb, but the tumor either never develops or becomes smaller. So if you knew that somebody was going to have a brain tumor, maybe not even to the 30s, but you could start giving them a drug in childhood, I think we all would do that. But should we go earlier? Let's go even earlier. We can go in utero. This is an example of taking stem cells, going into a fetus. This is a mouse fetus, that's blue dye over here. And as you can see, the cells, well, let me go back here. The cells participate in cerebrogenesis of the brain. Those are the blue cells. They're in the cerebellum, they're throughout the cortex. They become the various glial cells, oligodendrocytes, astrocytes with foot processes onto blood vessels. They become pyramidal neurons, non-pyramidal neurons that under EM receive synapses, make synapses, and the cells themselves are electrophysiologically active. Interestingly, you go in early enough, they will become part of the suprachiasmatic nucleus, which responds to light because it's part of the circadian rhythm. And I won't go into a lot of detail here, but to show that if you look for CFOS as a poor man's way of looking at activity, these cells will integrate with the host cells. So you turn on light onto the eyes, and along with the animal's own suprachiasmatic nuclear cells lighting up, so as you can see over here, does the donor cell. Well, that's a mouse. What about, let's start moving into a primate. Now this starts getting a little bit closer to something that we could actually do. This is an ultrasound of a fetal monkey. Here's the ventricle right over here. And a very simple procedure. It's a single needle under ultrasonic guidance into the ventricles. The donor human neural stem cells migrate up the radial glia. In fact, some of them do become radial glia, and they start participating in primate cerebrogenesis, as is illustrated over here by the red lines. And as you can see, this is a donor cell. This is a human neural stem cell integrated right into the proper cortical layer with a primate, with the monkey's own pyramidal neurons. And we can show their neurons through staining. What's interesting is, and also they become glia, oligodendrocytes and astrocytes, they also, early on, right during cerebrogenesis, start creating what are called adult neural stem cells. They get planted around the ventricles, but in utero, in fetal life, as you can see over here. So now, can we use this to start treating even an adult disease early on, or at least a postnatal disease? So Jean-Pio Lee did that with a lysosomal storage disease. She went in early in utero. This is called Sandoff disease, which is a deficiency in hexosaminidase A and B. If she goes in early, the cells integrate throughout the cortex, throughout the cerebrum, in fact, and extends lifespan and functional lifespan. Now, we can discuss why ultimately do the animals die, and it's probably because the mutant cells ultimately do take over, and they do it by different kinds of mechanisms, and I won't read them over here. But you could still say, well, that's a kid's disease. Well, what about if it weren't a kid's disease? What about if you decided to treat an adult disease in utero? And the one I'm thinking about is this one. We know when a fetus has Huntington's disease. If you bother to test, you can figure it out. And if in fact donor wild-type cells can integrate seamlessly into cerebrogenesis, you may not cure the disease, but maybe you can change it from a lethal disease into a mild or moderate inconvenience. So how about going earlier? Not just in utero when the fetus is already formed, how about actually at conception? And this is feasible. We're starting to do some clinical, not me, but I'm on committees that oversee some of this, mitochondrial diseases. Defective mitochondria are carried in the mom's chromosome. So what can be done, and there are clinical trials in the UK and in Australia talking about doing this now. You can take the nucleus of a mom from her oocyte where she has bad mitochondria, move it into an oocyte of a good donor, normal mitochondria, get rid of that oocyte's nucleus, put that in there, and now seminate that with the dad. And this is what we call three-parent babies, but that offspring will not have mitochondrial disease. And just in a dish, this is being done. Metalopov has done this, and again, we don't implant these yet, though clinical trials are going on right now in the UK and Australia. We all know about CRISPR, where you actually can selectively make gene modifications. And that's actually been done in adults. This is a paper that came out about 2 years ago, doing it in the liver, not the nervous system, but in the liver of this genetic cause of amyloidosis. I'm winding up now, but I'm going to end with the most controversial of all. I've led you completely down the garden path of what can be done and what can't be done. What about doing it here? Okay. Now, the further out here, you could say, well, that's just going way, way back to what you were talking about before. And you're right, that mostly will stay somatic, just in that particular offspring. But if you go back here, there's a chance, or at least theoretic, that it could be in the germline. It could be all your lineages, all your kids and your kids' kids forever. So is that an ethical dilemma? Well, I think this kind of stuff, I have to tell you, in my circle makes for absolute Talmudic debate. Where is your pain point? Now, to conclude, we can treat early in some cases if we know in advance that the defect exists or will exist, and if we have the right tools. The big question should be, should we, and if so, when, in whom, and at what cost? Not just financial cost, but sociologic cost and other things. And then there's some considerations that I know I had not initially thought about, but I've been forced to think about this when you start wanting to do precision medicine early on. Day of birth, maybe before birth. Is there such a thing nowadays, in the days of 23andMe and things like that? Is there true patient confidentiality? And can that data be co-opted by law enforcement to find out your relatives or other things that you've done? If we do get a drug that comes out of some of this work, who owns it? Who owns the IP? Who develops the drug? What about when can you consent a fetus? Is it good enough to consent the parents, Or do you have to reconsent the kid when he hits 18? And then what about if you start finding out things you don't want to know? The obvious one is finding out about diseases about which you can absolutely do nothing, even with the tools we have now. You can go in as early as you want, we just don't even know how to treat it. The other is things that maybe should stay in the family's closet. Anyway, I'm actually fairly optimistic that as long as we know about these, we can deal with them, because my feeling is that good science makes for good ethics, and I think, you know, in 50 years, this will be what we'll be doing. So I'd be happy to take any questions or debates or whatever.
That was brilliant, Evan. I'm going to call on Graham Parker, who gave us a lecture on ethics. And what was the most disquieting, Graham, to you in this presentation?
There was the beginning.
And then there was the middle. Oh, and the bit at the end, that was quite disturbing as well. Actually, Evan, fantastic talk. Thank you so much. I mean, we could we could take the stage and go 2-3 hours on what you've been talking about. But the one thing you didn't raise on that last slide, God bless America, is the issue of insurance companies.
Yeah. Well, that's sort of, yes, completely. And I alluded that a little bit, that I think routine obstetrical care should include starting to profile babies, certainly at birth and prenatally, and that, of course, can't be done with the American healthcare system now. And then, of course, you're totally right. Who's going to pay for all this, ultimately?
Who's going to pay for all this? And the second point I want to make, because I know that Alan Giacomo would want me to make it, is my new journal, Gene by Environment, because...
I'm sorry, the which?
My new journal, Gene by Environment.
Oh, okay.
Because I think what would be fascinating with what you find out when a child is born is, should that child live in Detroit? Should they be moving to Phoenix before the child has, even started to grow? You're opening up a whole slew of extra questions that responsible parents should be asking and answering for themselves. And people are going to make an awful lot of money off opening up clinics that would give the advice that those parents need.
Now, of course, we need more data. I mean, the more data you have, the more data you have to not be able to interpret. And I think many families have known, we've been doing amniocentesis for probably 50 years now. Most of the time you get back information that does not predict how your baby will look. It'll be magnified when we start doing this. I'm not saying we shouldn't do it, but what we should start doing is accumulating so much information and doing clinical correlations so much that we can figure out what are the important bits and what are the bits that just are part of normal variation or that get quality control, at birth.
Following on the question of who pays and is it ethical, shouldn't an insurance company want to pay for some of these treatments because they're going to save a lot of money? If you have a small payment, a relatively small payment at that stage in life, you don't have to pay much more money later on. Shouldn't governments, if they're enlightened, be paying for this? Because if people stay healthier for longer, then there isn't the costs of people being sick and other relatives having to give up their jobs to look after them. So maybe if we had the right attitude to this, we'd see there's an enormous longevity dividend. And to me, that would be the really ethical thing to do.
I can't agree with you more. I probably shouldn't be living in this country anyway, because I firmly believe in socialized medicine. I don't think families should be paying for any of this, quite frankly.
Any other questions? Professor Gorodetsky.
Thank you for the extremely interesting lecture, but we have to dive into the rationale of using stem cells. And people expected from stem cells to build tissue, to build organs, so forth, and it really hasn't materialized over the years. But things have developed. And the effect was mostly, in most cases, including some of your examples that you gave, were indirect. For instance, when you talk about mitochondrial disease, nowadays you don't have to put cells in order to replace the cells with the wrong mitochondria. Mitochondria can migrate from healthy cells, or you can even separate mitochondria and put them into the cells. So you don't need the stem cells here to replace the tissue. In most cases, they don't. Also, you have a lot of treatments with allogeneic cells that work fantastically. They don't integrate in the body, but they secrete. You talked about the secretome. They secrete whatever they need to secrete, like Kaplan would call it medicinal cells, whatever. He doesn't call them even stem cells anymore, even though he was a priest of the mesenchymal stem cells. So we have to be really careful to differentiate from our imagination of building new organs and so forth into the reality in which cells do have an effect, which in most cases is not replacing the cells, but actually enforcing the cells of the patient to act as we wish them to do.
No, I completely disagree. I completely agree with what you're saying. My view is that is all stem cell biology. So my view of stem cell biology is the same as developmental biology. I equate the two. And using stem cells for cell replacement is one tool when it's appropriate. There's certain diseases where it's appropriate. There's certain diseases where it's totally inappropriate or can't be done. What informs me all the time as to how one should approach a disease is what is the gap in therapy? And then I look to see what my tools are and whether a stem cell, can meet that gap. And what is it about the stem cell that meets that gap? And for that, I go back to what happens in development. You know, when there's a normal developing fetus, what are the cells doing? And if I think that I see a defect, whether it's secreting a various growth factor or creating an extracellular matrix, I look to see whether I can fill in that gap. So I completely agree, the old notion of stem cell biology or regenerative medicine being simply, replacing parts of your car is outdated now. But embracing a greater view of understanding development and then also understanding the disease. I think even as we try to understand better what the tools are, we should still never stop trying to understand what causes the disease. And we're constantly wrong there. Even when we think we know what needs to be fixed, we're always wrong. We thought ALS was a motor neuron disease. It's an astrocyte disease. We thought Parkinson's disease was a dopaminergic disease. It's probably not just a dopaminergic disease. It's probably an autophagic defect in other places. So we're on the same page there.
Evan, this will be our last question here.
Okay.
Hi. Professor Snyder, thank you very much for this very instrumental, inspirational talk. You have mentioned so many very sophisticated preclinical frontier works, we can show the disease being minimized and getting better for the animals. So just wonder if you could comment on for the route to do translational work, go from preclinical all the way to human trials, scale out and scale up for the treatment. What are some main challenges you see could be the roadblocks and what we should be working on? I particularly see biopreservation can be a major part of this because we've been using DMSO to preserving the cells. You mentioned epigenetic transcriptome and all those proteomic changes, this agent actually causes all of stuff. So the simple thing like that, if we continue using it, I see there's still some big challenge here. I just wanted to hear your comments on the...
No, I agree with what you said. People always ask me, what's the biggest challenge in regenerative medicine? Is it cost, regulation, funding, government, politics? And I still always say the biggest obstacle is the biology. And I still stick with that.
Thank you.
Thank you, Dr. Snyder. And let's give a nice round of applause to our esteemed colleague,